Welcome to Biligoloji, where we decode the latest breakthroughs in science. Today, we're talking about something that sounds like it's straight out of a horror movie, but it's happening in labs right now. We're talking about zombie cells and how science is literally resurrecting the dead.
You heard that right. I'm going to say it one more time just so it sinks in. Scientists have taken cells that were, for all intents and purposes, dead and brought them back to life.
So the big questions are, how on earth did they pull this off? And, more importantly, why is this a breakthrough that could literally change the world as we know it? That's what we're digging into today.
So let's get into it. This incredible story comes to us from the brilliant minds at the J. Craig Venter Institute, or JCVI for short. Now, if you know anything about synthetic biology, you know these guys are the absolute rock stars of the field.
And look, I want to be crystal clear here. This isn't some crazy theory or maybe one day kind of thing. This is a real, honest-to-goodness experiment with results published in one of the most prestigious science journals on the planet, Nature. This is the real deal.
Okay, so what exactly is a zombie cell? Let's break it down. You start with a normal bacterial cell. Then you completely shut down its own DNA, which basically makes it functionally dead. It can't do anything. But then, and this is the crazy part, you resurrect it by giving it a brand new synthetic set of DNA from a totally different species. This quote from Dr. Zimra Peksalam-Seidel, one of the lead scientists, just captures it perfectly, doesn't it? The cell is destined to die, but we give it life. I mean, wow. They're taking something that's on a collision course with oblivion and rebooting it with a whole new identity.
So, how in the world do they actually do that? Let's walk through the recipe. The best way to think about it is like a high-tech body swap, but for tiny little bacteria. It really boils down to three main steps. First, you have to inactivate the recipient cell. Basically, you kill its ability to use its own DNA. Step 2, you perform the transplant, inserting a completely new synthetic genome. And finally, step 3 is the magical part. You wait for the reboot. A tiny fraction of those cells actually accept the new programming and just spring back to life.
And it turns out there's a secret ingredient that makes this all possible. It's a drug called mitomycin C. It's actually a chemotherapy drug, and its job here is to just completely wreck the original DNA, leaving behind this perfect empty shell of a cell, just waiting for its new operating system.
Now, this is all incredibly cool, but to really understand why it's such a game changer, you have to know about the huge problem it solves. This wasn't just some neat party trick. It was about cracking a puzzle that's been holding back the entire field of synthetic biology for years.
Let's rewind a bit. Back in 2010, this same group made history by creating the first synthetic cell. But here's the catch. They had to put the new DNA into a living cell. After that, progress kinda hit a wall. Transfers were super unreliable and only worked between bacteria that were practically cousins. This new zombie method is what finally breaks through that wall.
The big, nagging problem with the old way of doing things came down to two words: false positives. Scientists thought their experiments were working, but really, the cells were just tricking them.
Okay, this is where it all clicks. With the old method, you'd put new DNA into a living cell, but the living cell could cheat. Its original DNA, which is find the one tiny piece of new code it needed to survive, say a gene for antibiotic-resistance, and stitch just that one piece into its own code. This is called homologous recombination. So the cell survives and it looks like a successful transplant, but it's a fake. But with the zombie method, the original DNA is dead. It can't cheat. The only way for that cell to survive is to reboot with the entire new genome. No more fakes, no more false positives. It either works or it doesn't.
So we've got the how, we've got the why. Now let's get to the really exciting part. What can we actually do with an army of resurrected cells?
Honestly, the possibilities are kind of mind-blowing. We're talking about designing custom microbes that can create biofuels way more efficiently. Or get this, turning bacteria into tiny little factories that pump out new life-saving drugs. We could build bacteria that are tailor-made to go out and clean up oil spills. And maybe most importantly, this gives scientists a foolproof way to test brand new genetic designs for important bacteria like E. coli, which could just supercharge the entire field of biology. To put it really simply, what these scientists have built is basically a universal USB port for life. It's a reliable, standard way to take almost any genetic software you can dream up, plug it into a piece of cellular hardware, and hit reboot to see what happens. It's a foundational tool for engineering life itself.
And that
of course, leaves us with the biggest question of all. We've gone from reading the code of life to writing it, and now to reliably installing it like it's an app. The question isn't, can we anymore? The question is, what should we create? Thanks for tuning into Biligoloji. Stay curious, and I'll see you in the next episode.